All right, so I want to make sure that you are ready for the titration lab in class. So I wanted to be able to finish up these notes and then do another video where I go through the calculations that you're going to do. So first off, as a reminder, titration is a lab technique. So you can do multiple different types of titration, and we're going to focus on different ones throughout the year. But the main thing is a titration involves the use of a burette, and we use those in the last labs. We're going to use them multiple times, but the key to a burette is that it basically allows you to add drop by drop very small measurable amounts that are very specific. It is way more accurate than a graduated cylinder or anything else. And so we can really get down to a very, very specific volume. And what we're going to use is we're going to use some substance where we know the molarity. So you are going to fill the burette with a known molarity solution. And then you're going to be able to see exactly what volume that was. And if you have volume and you have molarity, well, that's actually going to calculate and give you moles. So we're going to be able to figure out how many moles you need by using that volume and molarity out of the burette. So you're adding this very specific solution to some unknown. And most of the time, it's not wrong, it's just not what I meant to put right there. We're going to add it to a known volume of a different substance, and that different substance has an unknown molarity. There we go. So I'm going to end up knowing a volume, and I'm going to end up knowing a mole through a calculation, and through volume and moles, I'm going to be able to calculate a molarity of some sort of unknown substance. And this is the heart of titration, is it's basically a way to do stoichiometry in a lab. So you're going to see various different terms thrown around in problems and in labs. So the first one is standardized solution. And standardized solution just means this is your solution of known concentration. And we talked about this somewhat when we were doing dilution. So this is what you could think of as like your stock solution, all right? We know exactly what this thing is, and it's the standardized solution that goes in the burette. All right, so the standardized solution always goes in the burette. So the entire point of titration is to get to what we call the equivalence point. And the first part of the word equivalence implies equal. So then the thought is, well, what is equal? And what it means is that the moles of the two substances are equal. And you can't necessarily see this, but mathematically what you want is the equivalence point. So since you can't always see the equivalence point, we actually have something that's called the end point. And the end point, as implies, is when the titration is over. But how do you know when the titration is over? Well, you know when the titration is over because there is some visual change. Most of the time, that visual change is color. So there's a color change most of the time at the endpoint that tells you that the titration is over. And you want this to be as close to the equivalence point as possible. And to make sure you get a color change that is close to the equivalent point, you have to pick a proper indicator. So indicators are normally, it's some additional substance. All right, so it's an additional substance added, and we always add it to the unknown. And when we add it to the unknown, it's going to change color near the equivalence point. And there used to be questions that would ask you like to pick an indicator to make sure that you had a proper thing. That's not really the case anymore, so we're not going to worry too much about that at this point in time. So for titrations, we have two different types. The first, and what we will focus on primarily, is an acid-base titration. And acid-base reactions are neutralization reactions. So in neutralization, again, you have an acid plus a base, and you get salt plus water. All right, so you have a neutralization reaction, and it uses an indicator. So acid-base reactions, you have to have an indicator. There is no other way to do an acid-base titration unless you have an indicator. That is our most common type of titration. The other type of titration is an oxidation reduction titration, and this involves a transfer of electrons. That's what oxidation and reduction is. And this often involves transition metals. And the reason it's important to know that it involves transition metals is for two reasons. Number one, it is our transition metals, those guys that require the Roman numerals in the name. Those are the ones that can have multiple oxidation numbers, number one. And number two, transition metals change color. All right, so those transition metals that change color with different oxidation numbers. So frequently, you don't need to add an indicator with an oxidation reduction titration because the chemicals involved in the process itself, in the reaction, themselves will change color. So it's just kind of, we've been working with copper, and copper is typically blue. Well, there is a difference between copper 2+, which is normally blue, and copper 1+, which is normally green. And then we have other things like manganese will change between pink and purple. With iron, you're going to get ranges of yellow versus red. So you can get these different color changes with the different oxidation numbers of the transition metals. So that color change in and of itself tells you that you have reached an end point. So there is a general diagram in the chapter itself, but I just wanted to make sure that we were clear on what it is we were going to be looking at in the lab. So you are going to have a ring stand of some sort, and the ring stand is going to be holding your burette. And again, the burette, it's this long looking guy. It's got all the markings on it. It's got like a valve on there. And the burette is going to be filled, again, it's filled with known concentration. Okay, so that's where you're going to put the thing you know about. And then underneath it, you are going to have a flask of some sort, and that flask is going to be filled with your unknown. So it's going to have an unknown concentration, but it will have a known volume. Okay, and the number one thing that I'm going to say when you do this lab is don't forget to add the indicator. So where do we want the indicator to go? Well, we want the indicator to go in the flask, not in the burette, because I don't care if the stuff in the burette changes color. All I'm doing is removing things out of the burette. The flask is where the reaction is happening, right? The flask is where you are combining two different solutions. So that's where you need the indicator. Okay, so fundamentally, titrations are a specific type of stoichiometry problem. And stoichiometry means you require a balanced equation. Now, the nice thing is that neutralization reactions are almost always a 1-to-1, no coefficients needed to balance them. So it actually makes it pretty easy. You just have to have a good comfort level with molarity. So remember, molarity is moles over liters. And the other way you can think about it is moles over 1,000 milliliters, because these problems are often going to give you milliliter amounts. So if I have something that is 5.1 molar, I can rewrite that as 5.1 moles over 1 liter, or I could rewrite that as 5.1 moles over 1,000 milliliters. And I can use that as a conversion factor in my problems, which I will demonstrate in the next video.